In this lesson, we'll begin by looking at what enzymes are and why living organisms need them, then examine their structure and how they catalyze specific reactions, learn about the different digestive enzymes and where they're produced, and finally, follow food through the digestive system to see how enzymes break it down into usable molecules. Chemical reactions are essential for life. They build the molecules your body needs, break down food for energy, and allow your cells to communicate. But these reactions would be far too slow without help. This is where enzymes come in. Enzymes are biological catalysts produced by living organisms. A catalyst is a substance that speeds up a chemical reaction without being changed or used up itself. Living things have thousands of different chemical reactions happening simultaneously inside them. All these reactions need to be carefully controlled. One way to speed up reactions is by raising the temperature, but this approach has major limitations for living organisms. High temperatures would accelerate all reactions indiscriminately, including harmful ones. So instead, living organisms produce enzymes, specialized proteins that can speed up specific reactions at body temperature. This allows for precise control of which reactions happen and when. Enzymes are large protein molecules made up of chains of amino acids. These chains fold into specific three-dimensional shapes that are crucial for enzyme function. The most important feature of an enzyme is its active site, a special region where the chemical reaction takes place. The active site has a unique shape that allows it to bind to a specific substrate molecule, the substance the enzyme acts upon. Enzymes are highly specific. Each enzyme typically catalyzes just one type of reaction. This specificity comes from the precise shape of the active site, which only allows certain molecules to fit, similar to how a key fits into a lock. This lock and key model helps explain enzyme specificity, though the actual mechanism is slightly more sophisticated. When a substrate enters the active site, the enzyme adjusts its shape slightly to create an even better fit, a process called induced fit. Temperature and pH greatly affect how well enzymes work. Each enzyme has an optimal temperature and pH range where it functions most effectively. As temperature increases, enzyme activity initially increases because molecules move faster, creating more opportunities for enzymes and substrates to collide. However, if the temperature gets too high, the heat damages the bonds that maintain the enzyme's three-dimensional structure. This changes the shape of the active site, and the enzyme can no longer function properly. We say the enzyme has been denatured. Similarly, pH affects enzyme structure and function. Extreme pH levels can change the shape of an enzyme's active site by altering the chemical bonds that maintain its structure. Each enzyme has an optimal pH where it works best. For example, digestive enzymes in the stomach function best in highly acidic conditions, while enzymes in the small intestine prefer alkaline environments. Now let's see how enzymes are put to work in the digestive system, breaking down the food you eat into nutrients your body can use. The food you eat contains nutrients essential for life, but most of these nutrients are locked in large, complex molecules that your body can't directly use. These molecules must be broken down into smaller components that can be absorbed into your bloodstream with the help of digestive enzymes. There are three main types of digestive enzymes, each targeting a specific type of nutrient. Carbohydrases break down carbohydrates like starch into simple sugars such as glucose and maltose. Amylase is the main carbohydrate in your digestive system. It's produced in three different places: the salivary glands in your mouth, the pancreas, and the small intestine. Proteases break down proteins into amino acids. The stomach produces a protease called pepsin. Additional proteases are made by the pancreas and the small intestine. Lipases break down fats, also called lipids, into fatty acids and glycerol. These enzymes are primarily produced by the pancreas and the small intestine. Once these large molecules are broken down into their smaller components, sugars, amino acids, fatty acids, and glycerol, they can easily pass through the walls of your digestive system into the bloodstream. From there, they're transported to cells throughout your body where they can be used for energy or to build new tissues. While not an enzyme itself, bile plays a crucial role in the digestive process. Bile is produced by the liver and stored in the gallbladder until needed. It serves two important functions. First, food leaving the stomach is highly acidic due to stomach acid, but enzymes in the small intestine work best in alkaline conditions. Bile helps neutralize this acid, creating the optimal environment for these enzymes to function. Second, bile helps emulsify fats, breaking large fat droplets into tiny ones, similar to how dish soap breaks up grease. This significantly increases the surface area of fat exposed to lipase enzymes, dramatically accelerating fat digestion. The digestive system demonstrates the incredible efficiency of enzyme catalyzed reactions. Without these specialized proteins, we couldn't extract the nutrients we need from food. In our next episode, we'll explore another vital system, the respiratory system, and how it provides the oxygen needed for cellular respiration.